Hello dear learners, myself Dr. Shilpa Vinodia, Assistant Professor, Department of Botany, Pandit Sundarlal Sharma, Open University, Chhattisgarh, Bilaspur. Today we will continue with the practicals of BSc final year. In this module, we are going to start with the experiment separation of plant pigments using strip paper chromatography technique. Paper chromatography is a powerful and simple separation technique that has been used for many years in the scientific community. This technique involves separating a mixture of different substances by their movement along a paper strip. The movement of the substances is due to the differences in their solubility in a given solvent. For performing this experiment, we need fresh green leaves or colorful leaves, acetone, petroleum ether, gas jar, mortar and pestle, Whatman filter paper, etc. Paper chromatography is based on the principle of differential solubility. The stationary phase in this technique is a piece of filter paper and the mobile phase is a liquid medium. A small spot of the mixture to be separated is placed on the filter paper and the paper is then placed in a solvent. The solvent moves up the paper by capillary action, and as it moves, it carries the different components of the mixture along with it. The movement of the different component is based on their solubility in the solvent. The more soluble a component is, the further it will move up the paper. The less soluble a component is, the less it will move on the paper. The procedure for paper chromatography is simple and straightforward. The following are steps guided how to perform paper chromatography. The first step is to prepare the stationary phase. For this, we need to cut a piece of filter paper to the desired size. The size of the paper depends on the amount of mixture to be separated. The paper should be long enough to hang over the edges of the container holding the solvent. The second step is spotting the sample. In this experiment, we have taken fresh leaves extract of Tridex procumbens prepared in the mortar and pestle. Using a capillary tube or micropipette, spotted the same sample onto the filter paper. It should be noted that the spot should be small and concentrated. The spot should be allowed to dry completely before the proceedings. The third step is to prepare mobile phase. For preparing the mobile phase, 25 ml petroleum ether and 3 ml acetone has been taken in a gas jar, keeping in mind that the level of the solvent should be below the spot on the filter paper. Fourth step is placing the paper in the container. For this, we place the filter paper in the container with the solvent. The paper should be held in place so that it does not move. Then, next step is developing the chromatogram. It allows the solvent to move up the paper by capillary action. The solvent will carry the different components of the mixture along with it. Once the solvent has reached the top of the paper, remove the paper from the container and allow it to dry completely. Sixth and the last step is analyzing the chromatogram. The chromatogram can be analyzed visually or using other methods such as UV or fluorescence spectroscopy. The different components of the mixture will appear as spots on the paper. The distance travelled by each component can be measured and used to identify the component. Result: RF value can be calculated using the formula, that is, RF value is equal to distance travelled by the pigment on the stationary phase upon the distance travelled by the solvent. As you can see in the table, different plant pigments have different RF value and it can be compared from the standard values for confirmation. In this way, the unknown pigments can be identified in the samples. What all precautions we need to take while performing this experiment are as follows. First one is, petroleum ether and acetone ratio in solvent should be 25:3 only. Second, sample should be carefully loaded on the strip. Third one, only the tip of the paper should be in touch with the solvent and not the sample spotted. And the last one, but not the least, after the completion of experiment, the strip paper should be properly dried in air and the bands should be marked with pencil for better identification of bands. Chromatography techniques has many advantages, and it is simple, low cost, and can be performed with basic laboratory equipments which make it accessible to a wide range of users, including students and researchers with limited resources. The second one is rapid analysis. This technique gives rapid results, and this is time consuming also. So it provides rapid analysis of mixtures with results obtained within a few minutes. This makes it useful in clinical and forensic analysis. The next one is non-destructive analysis. Paper chromatography is a non-destructive technique, which means that the sample can be recovered after the analysis. This is particularly useful in cases where the sample is rare and expensive. The next one is high separation efficiency. Paper chromatography can separate a wide range of compounds with high efficiency. This is due to the high surface area of the filter paper and the ability of the solvent to penetrate the paper and interact with the compounds. So this was all about the experiment. Thank you so much for